Hello everyone. So this will be our last lecture for our biochemistry SI. In this lecture we will be covering two units. The first is the biosynthesis of amino acids via one-carbon carriers, and the second will be nucleotide metabolism. The first part I want to go over is the difference between essential and non-essential amino acids. So on this table on the left we see that there are 9 essential amino acids, and essential amino acids are amino acids that our body cannot synthesize by themselves, so we require them to be given by diet. So we see on the right there's this graph that indicates the difference between essential and non-essential amino acids. So in magenta we see that these essential amino acids, those 9 over here, have a lot more steps required in their biosynthetic pathways compared to these blue non-essential amino acids. So this explains why we need to have these amino acids by diet, because our body doesn't have the capabilities to do these 9, 11, 12, 16-step pathways to create them. That's just way too much work. So we'd much rather just get them through ingestion. And we see that this makes sense because these amino acids with weird structures like tryptophan or phenylalanine, methionine, these really weird structures that would be hard to make synthetically are our essential amino acids. So our amino acids actually play a very important role in the biosynthesis of new compounds within our cell. In other words, anabolism. And there's an important intermediate in this process called a one-carbon carrier that strips off one-carbon fragments from these amino acid backbones and adds them together. So the two one-carbon carriers we'll be looking at are THF and SAM. So THF is short for tetrahydrofolate, whereas SAM is short for S-adenosylmethionine. So we see here THF has 3 main domains that we'll be going over, whereas SAM has a nucleotide resembling part as well as an amino acid chain on the left. So this is a more in-depth look at what THF looks like. So we see that there are 3 parts: the pteridine, the para-aminobenzoate, also known as PABA, P-A-B-A, as well as glutamate or polyglutamate residues. So the pteridine, it's basically 2 heterocyclic rings that have nitrogen groups in them, and it gets its name pteridine from the Greek word for wings, because this part was initially a pigment discovered in butterfly wings. So that's where the name comes from. And PABA, para-aminobenzoate, is an important ingredient of sunscreen that blocks UV radiation. And that makes sense because it has conjugation, so you would expect it to interact with UV radiation. And on the right over here we have either one or several glutamate residues, and this is actually found in spinach, so raw spinach is a great way to get these glutamate residues to form THF. But if you cook this raw spinach and overcook it way too much, then you'll essentially destroy these glutamate chains, so they won't be useful. So the moral of the story is you should eat raw spinach rather than overcooking it. So let's look at the THF synthetic pathway. So on the left here is the pathway in bacteria, and on the right is the pathway in humans. Let's examine the one on the right first. So we see here that humans ingest folate. So if you look on any nutrition box, like let's say your cereal, you'll see there's something for folate or folic acid. And that's what this is, because humans can ingest folate by themselves. So they'll take this folate, and they have an enzyme called Dihydrofolate Reductase, DHFR. So what that enzyme will do is it catalyzes 2 successive reduction reactions. So the first one, folate will be reduced by NADPH, which is a good reducing agent, and that'll form dihydrofolate. So between these two names we saw that we added on 2 hydrogens here, which is what reduction is, and we see it's over here in pink. And now a second subsequent reduction will be catalyzed by the same enzyme, dihydrofolate reductase, and that'll create tetrahydrofolate. So the 2 hydrogens now added on are on the top. So we see that to get from folate to tetrahydrofolate, we just need to have 2 reductions. However, in bacteria, there's something different going on. And the interesting thing here is bacteria actually cannot absorb folate from the environment. So humans have it easy. They can take this and just 1 enzyme, also 2 reactions, you're done. Here it's a bit more complicated. You actually have to synthesize your own folate. So bacteria will start off from PABA and pteridine. Remember those 2 parts up here, right? This was PABA and this is pteridine up here. So they'll take these two and they'll use an enzyme called Purine synthetase, which requires ATP, to form dihydropteroic acid. And then this will undergo another reaction to form dihydrofolic acid, which will then enter in this same part of the pathway. So essentially, what we produce over here, dihydrofolic acid, is basically this part. So now it can sort of override this step and then continue off to form THF over here via the same enzyme. So in humans, this dihydrofolate reductase will catalyze 2 reductions, whereas in bacteria only 1, because you can't absorb folate from the environment. You have to go another route. Interestingly, there are 2 drugs that are pretty important in this pathway. One of them is Methotrexate, and the other one is Sulfonamides. So Methotrexate works in both bacteria and humans because it inhibits this last enzyme in the pathway, which is dihydrofolate reductase, that catalyzes these reduction reactions. So essentially, if you add a methotrexate, you'll prevent this 1 carbon carrier from being formed. The other drug is sulfonamides, but this only works in bacteria and not humans. And the reason why is that it inhibits one of these earlier steps, Purine synthetase, which isn't really present in humans because we already get folate from the environment. We don't have to make it ourselves. Alright, so let's first take a quick look at the sulfanolamids. So in this case, we see here that this is the PABA ring that was part of the dihydrofolate, right? So this red part over here is the PABA, and a sulfanolamide looks like this. So we see it has this conserved benzene ring, it has the amino group, and paired to it, instead of a carboxylic acid, it has some sulfur acid conjugated to it. So we can see they're very similar, and this is important because sulfanolamides actually serve as PABA analogs that can inhibit this purine synthetase. So normally this enzyme over here, it binds PABA and puts it together with purine. But we see here sulfonamides, they resemble PABA very well, as seen over here. So they can actually take the place of it and competitively inhibit this enzyme. So an interesting thing, though, to remember is this only works in bacteria and not humans. So sulfanolamides, they don't harm humans. They only harm bacteria. The other drug that I mentioned was methotrexate. So methotrexate is a dihydrofolate reductase inhibitor. And remember, this enzyme is the sort of the last step in this THF pathway. So it's found in both bacteria and humans. So this drug works in both bacteria and humans. We see here that dihydrofolate has a structure with the pteridine, PABA, and the glutamate residues over here. And methotrexate resembles that very well, except for this methyl group on the nitrogen, and this carbonyl turned into an amino group. So once again, methotrexate also serves as a competitive inhibitor by sort of acting as dihydrofolate and binding to this enzyme and inhibiting it. So this is pretty interesting because a lot of cancer patients are actually prescribed methotrexate. And the reason why is your 1-carbon metabolism, like I mentioned before, is very important for building things. So your THF, your 1-carbon carrier, it's very useful for making nucleotides and DNA and RNA. And we know that cancer cells are cells that divide way too quickly, right? They form tumors, which are just big balls of cells because they grew too quickly. So they need to be able to replicate their genome to keep up with this. However, if you don't have a replete supply of nucleotides to propagate this, then your cancer cells will die. So that's where methotrexate comes in. Methotrexate inhibits 1-carbon carriers like THF from being produced, right? Because it inhibits this DHFR enzyme. So now without THF, you can't produce your nucleotides because you need to add 1-carbon pieces to create them. Now you can't divide and your cancer cells will die. So here's a cool diagram that sort of explains the process. So we start off with 4 main amino acids. We have serine, glycine, histidine, and tryptophan. And these 4 amino acids can donate a 1-carbon fragment to THF. So remember, we just saw how to produce THF via those synthetic pathways. So now we have it. What do we do with it? Well, it'll react with these amino acids. And remember, urea comes off, which can then enter the urea cycle because the nitrogen isn't useful, right? But it is toxic, so we need to neutralize it. But the THF picks up the good stuff, which is the carbon. So now we have THFC1, which basically means THF with the carbon on it. Now this compound can go on to do big things. So there are three main pathways that this can go towards. First, it can be used to make purine bases through the de novo Purine synthesis pathway. Second, it can be used to make thymine, which is a deoxynucleotide. And third, it can be used to make S-adenosylmethionine, SAM. Now, if you guys remember, in this slide up here, slide 4, I told you guys that SAM was the other one-carbon carrier. So this is an important thing to note. These guys aren't two separate things. Your THF is actually required to make the SAM cycle work. So these two are inextricably linked. So now, if you produce the SAM, which is sort of the more interesting pathway, this can then go on to do a whole bunch of things, like produce phosphatidylcholine, important for membranes, creatine, epinephrine, which is a neurotransmitter, melatonin, important for sleep, DNA methylation, important for gene regulation, and methylation of drugs, which is sometimes problematic because then your drugs won't work properly, so they need to be tested against that. Alright, so here's a quick question for you guys. Feel free to pause the video because I'll be going pretty quickly through it. Which is not a component of THF? Pteridine, sulfanolamide, PABA, or polyglutamate? Alright, so the answer to this question is sulfanolamide. So remember, the pteridine was found in the butterfly wing pigment. That's how it got its name. PABA, para-amino benzoic acid, was a component of sunscreen that blocks UV radiation because it's highly conjugated. And I mentioned the polyglutamate was found in raw spinach. Your sulfanolamide, remember, was a enzyme, it's a drug that actually works only in bacteria by inhibiting DHPS, the pteridine synthetase enzyme. And it competitively does this by resembling PABA, because it had that same benzene ring. Alright, which two of these are one-carbon carriers? Okay, so the answer to this one is THF and S-adenosylmethionine, also known as SAM. So your folate is just an intermediate to create THF, same with your dihydrofolate. Your serine actually donates a carbon to THF, so none of these are one-carbon carriers, only THF and SAM. Alright, here's a true and false question. Sulfanolamides only work in bacteria. True or false? So this answer is actually true, because remember, sulfanolamide is a PABA analog that inhibits pteridine synthetase. This enzyme is only found in bacteria because bacteria cannot make their own folate, so they have to synthesize it de novo rather than taking it from the environment like humans do. Methotrexate works in both bacteria and humans. True or false? So this one is actually true. Methotrexate is a dihydrofolate analog, which means they have similar structures. So it's able to fool this enzyme DHFR, which was dihydrofolate reductase, the last step in that pathway to create THF. And this enzyme is actually found in both humans and bacteria because it's the last step. So methotrexate will inhibit it in both humans and bacteria. And remember, this is a novel cancer therapy by inhibiting cancer's ability to replicate because it inhibits nucleotide biosynthesis. Which of these is a cancer treatment? Alright, I guess I gave that one away. So yes, this one is methotrexate, because remember, THF and other one-carbon carriers are important for nucleotide and DNA synthesis. And cancer cells really need to replicate their DNA a lot quicker than our normal cells. So if you inhibit one-carbon metabolism, you, by extension, inhibit DNA replication. Which of these donates a carbon to THF? SAM, urea, alanine, or serine? So in this case, it is serine. Your SAM, remember, it actually gains a carbon from THF, so it doesn't donate it to THF. THF is upstream of SAM. Urea, that's just a byproduct of amino acid metabolism. Alanine, that could be a trick question, but it's not alanine. It's actually serine is the most common amino acid that can donate a carbon to your THF. So remember, the other three are glycine, histidine, and tryptophan, but serine is the most common. Which of these is not a biosynthetic destination of THF? Remember, I told you there were three, so one of these is not the case. All right, so this one, your THF is responsible for a lot of nucleotide metabolism, right? So it's used in de novo purine synthesis, as we'll see later in this unit. It's used to create thymine, which is a deoxynucleotide, and it's used to create SAM. However, the DNA methylation is actually catalyzed by SAM, so it's not THF in this case. Okay, so we talk a lot about THF's ability to donate carbons, but what does it actually look like when it gains one? So in this case, we have our THF over here, and to put it into perspective, there's like another ring over here. So these are our two heterocyclic rings that form the pyridine moiety. And down here we have our PABA, and the glutamates are down here as well. But really, the important part where we gain a carbon is the nitrogen in the pyridine and the nitrogen in the PABA. So you'll gain this carbon in between them. But there are 6 ways actually this carbon can take form. So let's look at them. One important thing for nomenclature is that nitrogen in the pyridine ring is number 5, and the nitrogen in the PABA is number 10. So let's start from the left. So over here, there's a carbon that's attached to both of these nitrogens. So we write N5 for this one and N10 for this one. But however, this one is named methylene. So anytime you see a CH2, this is named methylene, as opposed to CH3, which is methyl. So because it's CH2 and it attaches to both N5, N10, methylene. The second one over here is just the CH3, which I just said was methyl, right? However, this one only binds to this nitrogen over here, which is the 5th one. So this is N5 methyl. This third one over here, this carbon actually is oxidized. So we see it's sort of like an aldehyde, and it's bound to this nitrogen on the right side, which is number 10. So this is N10, but instead of calling this an aldehyde, we say formal. So this is N10 formal THF. So anytime you see an aldehyde, think formal. Over here, we have something weird. So before we saw CH2, and we saw CH3, but here we just see CH. So this is called methanil. So make sure you guys keep this in order because it can get confusing. So methylene, methyl, methanol. They're all very different. So in this case, there's only one hydrogen, and there's a double bond to N5 and a single bond to N10. So it's N5, N10, methanil. In this 5th form, we see that we have this carbon. It's oxidized, but it's not attached to an oxygen in this case. It's attached to a nitrogen. So this is a form amino, and it's attached to this 5th nitrogen up here. So this is N5 form amino THF. And this last one over here, we see an aldehyde. Remember, I told you guys that was formal. So in this case, it's attached to N5. So it's N5 formal compared to this one, which is N10 formal. So you see in this case for formal, it can appear on either one of these nitrogens. So you have to really specify the nitrogen it appears on when naming it. You do have to know these 6 for the exam, and I will test you on them right now. So just keep that in mind, okay? Okay, so we just saw how THF looks when it accepts a carbon, right? So this is THF, and this is THF when it has a carbon, in this case methylene, because of the CH2 over here, right? But where does it get this carbon from? So like I mentioned before, there were 4 amino acids, but serine was the most common donator of a carbon group. So in this case, there's an enzyme called Serine Hydroxymethyltransferase that takes serine, remember this is the alpha carbon, COOH and amino in the backbone, and then in the R group we have a CH2 connected to an OH, so this is serine. This carbon will actually pop off from the backbone to form a formaldehyde, which will then be attached to THF to form methylene. And this is now sort of the C1 THF form. It's the THF with the carbon. But what happens to serine? Well, it loses this carbon and oxygen, so all you're left with is a hydrogen. So that's going to make it glycine. So now, as a byproduct of serine giving up its carbon to THF, glycine will be formed. And the cofactor for this reaction is PLP. So when your serine donates, so here's our serine from before, when it donates a carbon to THF, you're going to be forming N5N10 methylene THF. Remember, this is the CH2 form, so this is very important to know that serine won't cause any of the other 5 forms. It'll only cause this one, the methylene form. And remember, the byproduct of that is glycine, which can then also donate a carbon and form methylene THF as well. But the interesting thing to note here is that when we form our N5N10 methylene, it's not going to have to stay that way. It can also interconvert between all these other forms via oxidoreduction reactions. So it can turn into N5-methyl, methanol, formyl. It just has to undergo an interconversion reaction. And we see that these other amino acids, tryptophan, histidine, they also form other forms of THF over here. And we see, remember the 3 biosynthetic paths I mentioned before for THF. Number 1 was purine bases, so that's formed by the N10 formal THF. The thymine, which is the deoxynucleotide, formed by methylene. So we can see that's from the serine directly. So getting a carbon from serine will be important for forming thymine. And then the other form was SAM, which is an analog of methionine. Alright, so time for some questions. Which enzyme catalyzes the transfer of C1 from serine to THF? Remember, pause the video because I'll try and go through them a bit quickly. So in this case, it's Serine Hydroxymethyltransferase. Remember, this enzyme will cleave off the carbon and oxygen from serine, which produces a formaldehyde. And this formaldehyde is quickly captured by THF, which is important because formaldehyde is very bad. And in this process, serine is cleaved into glycine. So which form of THF is created by the serine hydroxymethyltransferase enzyme? Remember, these are the 6 forms we went over. Alright, so in this case, it is N5N10 methylene THF. So remember I told you guys in this diagram up here, we had serine, and it directly produces the methylene form, which was the one that looks like this, just the CH2 in between. So this is produced by serine. This is very important to know. And we see here in this reaction, the serine hydroxymethyltransferase will take off this part of serine, and the formaldehyde will be captured by THF. Oxygen will leave as water, and we'll be left with a CH2 that attaches to both the nitrogens. So we get N5N10 methylene THF. Alright, now let's try and identify some of these forms. So what's the name of this THF form? Alright, so this one, we see that we have a CH2 here, and it's attached to both N5 and N10. So this will be N5N10, so you can immediately cross out these guys, because they don't have N10 in there. This says methanol, this says methylene. So remember, methylene is CH2, whereas methanol was the weird one, where it was like a carbon with only one hydrogen, and a double bond on one side, and a single bond on the other. But methylene is the more important one, produced by serine. Alright, what's the name of this THF form? Alright, so in this case, we see we have a CH3, which is a methyl, and it's attached to the nitrogen in the pyridine ring, so this is N5. So in this case, it would be N5-methyl THF. Alright, what's the name of this THF form? So in this case, we have carbon with only one hydrogen, a double bond to N5, and a single bond to N10. So this is N5, N10, methanol, THF. So remember, this nomenclature can get a bit tricky, so make sure you guys stay on top of that, okay? And what is the name of this THF form over here? The last one, I promise. Okay, so in this case, we have a carbon that's oxidized to an aldehyde form. So this is called a formyl, and it's attached to this nitrogen in the PABA. So this is N10. Remember, the formyl group can either be on N5 or N10, so we really have to specify in this case. So here, because it's on N10, it's N10 formyl THF. Alrighty, so now that we got that out of the way with, let's see where we are in this diagram. So remember, from serine, we can transfer a carbon via serine hydroxymethyltransferase to THF. That THF will grab that formaldehyde to form N5N10 methanol. Was it methanol? Methylene, THF. And that has three biosynthetic destinations. It has purine bases, thymine, and SAM. But SAM is the more interesting one. These other two, the first two, we'll be looking at in the next unit down below. But first I want to look at SAM because it has 6 different functions over here, so it's pretty important. Alright, so let's look at the SAM cycle. So remember, we just created our THF that has a carbon on it. So this is actually very important for propagating the SAM cycle, which consists of 4 intermediates. So if we start from a more common molecule, we have methionine. Remember, is the initial amino acid that's found in almost all proteins, encoded by the AUG start codon. So what's going to happen to methionine is it's going to grab an A, which is an adenosine. So it's just going to grab that base as well as the sugar, so the nucleoside form. And this is going to form SAM, so S-adenosyl methionine. So the methionine is constant, but this S-adenosyl is what gets added on. The S merely just means this adenosyl is added to the sulfur of methionine. So this stands for SAM. So now SAM has a methyl group that's highly reactive. So it can donate this methyl group to any precursor and form a methylated product. And in this process, it'll lose that methyl group once it's donated, and it'll be turned into SAH, S-adenosyl homocysteine. Now, SAH is not really useful, so we're going to cleave off that adenine and ribose sugar that we had added on in this step. So we just hydrolyze that off, and we're going to be left with homocysteine. Homocysteine is something you guys have never heard of before, but from the name you can tell it's analogous to the amino acid cysteine, right? So it's probably an amino acid, but it's just not something we use in the body. So we need to revert it to something more useful. So in this case, we revert it back to methionine to close the cycle. And this step where we methylate homocysteine to form methionine requires the THF we produced earlier on. So this is how THF is linked to SAM. Alright, so let's look at this step by step. So on the bottom left, I have that diagram I just showed you guys. So we'll be starting with this familiar molecule, which is methionine. So on the left here, we have methionine. Remember, this is the alpha carbon. We have our amino group and our carboxylic acid, which forms this backbone. 2 carbons in the R group, a sulfur, and then a methyl group sticking off the sulfur. So that's the classical methionine structure. And an ATP will come in. Normally with ATP, usually something like ADP falls off, or AMP. In this case, this is very different and very unique, so please take note to it. It's a very easy test question for someone to ask. Is 3 phosphates fall off. So remember, this ATP, we have this ribose sugar over here, right? It's a pentagon because it's 5-membered. We have our adenine base. I'll just draw it like that. And then we have 3 phosphates attached to the 5th carbon. So in this case, instead of cleaving it off here or here, we actually cleave it off here. So what that leaves us with is this ribose sugar as well as this adenine base, which is in blue over here. So this is the ribose sugar. Remember, this is not deoxy because we have an OH on the 2nd carbon, so it's just ribose. And this adenine base, which has this amino group sticking off. And we see that this this group, this fifth carbon, is attached directly to the sulfur in the methionine, which is why we have this prefix S. So this adenosyl in blue is attached to this methionine at the sulfur, so this is called SAM. And interesting thing to note is that when we create SAM, the sulfur gains a positive formal charge. And remember, from organic chemistry, formal charges are inherently unstable. So we just created an unstable molecule. Let's see what we do with that in the next step. So here we have our SAM we just created by taking methionine and adding on the adenosine nucleoside. So in this case, we have a positive charge on the SAM, which makes it unstable. So something wants to come off of it to relieve it of that charge. In this case, that will be this methyl group. So something will come along, it doesn't matter what, and it's going to pick up this methyl group. And what you'll be left with is this compound with this bond cleavage. So this is called S-adenosyl homocysteine, SAH. This is basically SAM without the methyl group sticking off. And an important thing to note about SAM here is it's different from THF. We saw from before that THF had those 6 forms, right? It had the methyl, methanol, methylene, formamino, formal. So it had all those 6 different forms. However, SAM can only donate a methyl group, whereas THF could donate any one of those 6 types of groups. And an important thing to know is SAM can only methylate at OH or NH2 residues. And the reason why this methylation is possible is due to this positive charge on the sulfur. So this is a very easy question for someone to ask you, and you can say the reason why SAM can methylate is due to the inherently unstable positive charge on the sulfur. So yeah, we see here SAM that we just formed methylates this precursor, and you'll be left with S-adenosyl homocysteine, which is basically the methyl group chopped off. Alright, so now, like I said before, S-adenosyl homocysteine, SAH, is very useless, so we'll just cleave off this adenosine group so it can be remade into ATP later. So we use H2O and hydrolyze it, and what we'll be left with is homocysteine, so we cleave it at this bond over here. So if you look at homocysteine, it's basically, methionine would look like this when you have an extra carbon attached to the sulfur. It's basically methionine without the carbon. But interestingly, it's actually named after another amino acid, and that's cysteine. So let's see why. So here on the left we have cysteine. So remember, this is our alpha carbon, we have our amino, and we have our carboxylic acid up here. And for the R group, we have 1 carbon and then a sulfur. And remember, this can form your disulfide linkages. Your homocysteine, however, which we just produced, actually has 1 extra carbon in the R group. So because of that, it resembles cysteine very closely, so we call this homocysteine. So that hopefully will help you remember what the structure of homocysteine looks like. Interesting thing to note is that a study done in Sweden many years ago showed that homocysteine in high levels is actually bad for your body. Specifically, it causes hardening of your arteries and other CVDs, cardiovascular diseases. So it's very important that your SAM cycle keeps going round and round, because if there's a pause and you accumulate a lot of homocysteine, then that can lead to cardiovascular disease. So that's why a folate deficiency, right? Because a folate deficiency in humans means we can't produce THF. And remember, THF is important to methylate homocysteine back into methionine. So without that, homocysteine will accumulate and you get a lot of cardiovascular disease. So let's see this last step here. So we have this homocysteine, which is basically cysteine plus a carbon. So we're going to get our THF, and in this case it's going to be N5-methyl THF that we'll be using. So this N5-methyl THF will donate this carbon that's sticking off of N5, the pyridine ring, and it'll add it on to the sulfur of homocysteine to produce methionine. And in the process, it'll be reverted back to THF. So this is important because remember, homocysteine is bad for you. So we need this THF form to revert it back to methionine. If we have a folate deficiency, then you're screwed, because now you'll have a lot of homocysteine accumulating. And an important niche detail to know is that this reaction is dependent on methylcobalamin, which is vitamin B12. And the only other reaction in the human body, I believe, where this cofactor is used was back in the fatty acid chapter, where it converted methylmalonyl-CoA into succinyl-CoA. Alright, so that was a lot of material we just covered, so let's put it all together. So let's start from the top left and work our way. So remember, our humans can ingest folic acid. So it's pretty easy from this step onwards. We need to do 2 successive reductions via NADPH, and this will turn our folic acid into tetrahydrofolate. And this is catalyzed by the enzyme Dihydrofolate Reductase, which is found in both bacteria and humans. This enzyme can be inhibited by methotrexate. Remember, methotrexate is a common cancer drug because when you inhibit one-carbon metabolism, by extension you inhibit nucleotide metabolism. And cancer cells need to divide quickly, so they need to be able to replicate their genome. If you don't have nucleotides, then you can't grow. So now this THF needs to be carbonated. Probably not the right word to use, but essentially it's going to gain a carbon from serine, or one of these other 3 amino acids we mentioned, and in this case it's via the enzyme Serine Hydroxymethyltransferase. So this formaldehyde group of serine will come off, and THF will immediately capture it, and this will form N5N10 methylene THF. So remember, this form can interconvert between all of them, and this form specifically, I believe, was used for thymine, to create thymine nucleotides. But in this case, we're not interested in that. So we're going to interconvert this N5N10 methylene produced by the serine into N5-methyl THF. And now this N5-methyl can use a vitamin B12 dependent reaction to methylate our homocysteine, which is, remember, it's cysteine plus an extra carbon over here, and add on a methyl over here to form methionine. So now remember, methionine, which is a familiar molecule you guys should remember, will react with ATP. In this case, the 3 phosphates will actually fall off, so all you're left with is adenosine, which is the adenine base plus the ribose sugar. So this is a nucleoside, when you just have the base and the sugar and not the phosphates. So this blue part is the nucleoside over here. We have the ribose sugar and we have the base, and that's attached at the sulfur, producing S-adenosylmethionine, and this positive charge is extremely unstable, which allows SAM to donate this methyl group to any precursor. In this process, SAM loses this methyl group, and you form S-adenosyl homocysteine. This is pretty useless, so you're going to hydrolyze off this nucleoside base so it can be reformed into ATP or whatever. And in the process, you're left with homocysteine, which, remember, is very dangerous. It creates cardiovascular disease. So you really need this step to work at a high efficiency to convert it back to methionine. If you have a folate deficiency, all of this will come to a halt. Then you won't be able to form methionine, and a lot of homocysteine will accumulate, and you'll have cardiovascular disease. So hopefully this was able to show you guys how one-carbon metabolism works. Something interesting I want to tell you guys about was a 2-step authentication mechanism found in bacteria. So if you guys remember from genetics that in bacteria, the first amino acid is usually, it's not methionine, but it's N-formyl methionine, so they call it F-met. And this is actually a sort of 2-step verification that our cell is healthy enough to produce proteins. And these 2 steps are really to see if one-carbon metabolism is working. So let's see how. In the first step, we know that, well, this is a methionine, right? A methionine amino acid, and methionine is a very important part of the SAM cycle, as we saw. So if there's enough methionine to produce F-met, then we know that the SAM cycle is working properly. So that's our first green light. The second green light is, oh, this F over here. That means formal. And remember, your THF can form the N5 formal or N10 formal forms. So enough of this formal group to produce F-met indicates the THF cycle is also working properly. So when you know these 2 things, SAM and THF are good, then you can say, okay, I'm good to produce proteins. So this F-met is really a double test of your one-carbon metabolism. Alright, time for some questions again. How many high-energy phosphoanhydride bonds does SAM synthetase break? 0, 1, 2, 3, or 4? So in this case, remember we cleave off 3 phosphates when we add ATP, and we are left with this adenosine nucleoside that can be added onto methionine. So we cleave off these 3 phosphates, and the resulting nucleoside, which is adenosine, will be added onto methionine. So we need to cleave off 3 phosphates. So this is a very unique reaction. Which feature of SAM allows it to donate methyl groups? Is it the adenine nucleotide, the ribose sugar, the positively charged sulfur, or the Zwitter ionic peptide backbone? So in this case, the positively charged sulfur is the reason why your SAM can donate a methyl group. So we see here the SAM that we just formed, it has this positive charge on the sulfur from the methionine. So this means it's inherently unstable. So for us to alleviate that charge, it'll have to kick off one of these 3 groups. In this case, the thermodynamics just work out that the methyl will be kicked off. So the methyl will be added on to this guy, and you'll form S-adenosyl homocysteine. Alright, what converts homocysteine into methionine? THF, N5-methyl THF, N5-N10 methylene THF, or hydrolysis? Alright, so in this case the answer is N5-methyl THF. So remember, towards the end of the cycle, let's zoom in, we have a lot of homocysteine accumulating. However, this is really bad for us, so we need to add on a methyl here to form methionine. So the methylene, N5-N10 methylene THF we created from serine actually won't do the trick. The N5-methyl THF will, where we have a CH3. So we actually need to convert that methylene, that CH2 form, into the CH3 form, which can then do a vitamin B12 dependent methylation reaction on homocysteine to produce methionine. So yeah, in this case, you need the methyl form. Okay, so I can't ask you guys if you have questions, so I'll move on to the next thing, which is Chapter 25, Nucleotide Biosynthesis. Before we get into it, I realize a lot of people struggle with the nomenclature and really the structures underlying this chapter, so I want to make sure we have the foundation done well. So the first thing to know is, what is the difference between a nucleoside and a nucleotide? So let's look at the more familiar one first. Nucleotide is like ATP. You have your nitrogenous base, which would be adenine. You have your pentose sugar, which could be either ribose or deoxyribose. And you have your phosphate or phosphate groups. So it could be multiple, it could be 1. For ATP, there's 3. And for a nucleoside, it's basically the same thing as a nucleotide minus the phosphate group. So you're just left with this nitrogenous base and this pentose. And remember, this adenosine nucleoside was what was added onto methionine to form SAM. So this is very important physiologically. And let's look at these 5-carbon pentose sugars a bit more. So we see here they can either take one of 2 forms. They can either be ribose or deoxyribose. If we remember from the Fischer projections, ribose was a aldose sugar, so we had a CHO on the top, and I believe it was 3 OHs all to the right. And at the bottom there was the CH2OH. Like this. So we see here that these 3 oxygens, what happens to them? Well, one of them will be over here, the other one will be over here, and I believe the third one is over here. So, actually no, that's not correct. Let's see where they went. We know this oxygen acts nucleophilically to attack here, so that'll form a 1, 2, 3, 4, 5 carbon backbone. So this aldehyde will become your anomeric carbon. So this oxygen up here will be this oxygen. Now let's change colors a bit. This oxygen down here, so we'll go to the right. So that'll be this oxygen over here. And then the next one is this one, and the last one is, oh yeah, I actually was right. So it's this third one over here. So these are the 3 oxygens formed in the 3 carbons in the middle, and this last CH2OH is this thing sticking off over here. So that's ribose. Deoxyribose is basically the same thing, except at the second carbon there is no OH. Rather, it's replaced by a hydrogen. So this doesn't make sense really chemically, but something you read in a lot of papers is that this OH at the second position actually makes ribose more labile. Labile is a fancy word for saying it's easily degradable. So remember, your ribose is used to make RNA. Your deoxyribose is used to make DNA. Your RNA is just a very transient thing to get from the nucleus to the cytoplasm. Your DNA is really what's going to stick with you for life, right? It's in the nucleus, that's your genetic code. So really you want the deoxyribose to be more stable, so it's less labile, which it doesn't have the OH. So this is good. So not having the OH makes it more stable. Alright, so now let's look at the nitrogenous bases. Remember the two classes are purines and pyrimidines. Purines are double-ringed, pyrimidines are single-ringed. We see here that the two purines are adenine and guanine. Adenine just basically has an amino group on the top, whereas guanine has a carbonyl on the top replacing that, and the amino group gets moved to the bottom left. There are two pyrimidines in DNA, that's your cytosine and thymine. Your cytosine has an amino group sticking up and a carbonyl on the bottom left, whereas your thymine replaces that amino group with another carbonyl, so there will be two. And in addition to that, your thymine also has a methyl. Your uracil, however, is a pyrimidine found in RNA. So uracil is actually the very same as thymine. It has the two carbonyls, one up and one to the bottom left, except for this methyl group, which is replaced by a hydrogen. So the main takeaway is the difference between uracil and thymine, which are really interchangeable as they both bind with adenine, is that thymine has this methyl group, which allows it to be detected by your DNA polymerase enzyme. Alright, so now that we got the basics out of the way, there are two main classes of pathways we'll learn about. The first is the de novo pathway, and the second is the salvage. So de novo means essentially from scratch. You're creating these nucleotides out of single carbon fragments and amino acids. So amino acids and PRPP, which we'll later see what that is, but that's de novo. Your salvage is a bit of an easier pathway. It's not as energy consuming because half the work is already done for you. And by this, I mean you don't need these amino acids. Rather, you already have the nitrogenous base formed. And this can either be through degradation of RNA, so you recycle it, or it can be through your diet, where you ingest nucleic acids and you have an abundant supply of nitrogenous bases, which can then be incorporated with the sugar and phosphate groups to form nucleotides much more easily than having to create that nitrogenous base via the de novo pathway, which is more energy costing. So let's see what this PRPP is. This requires a bit of a throwback to the pentose phosphate pathway. So I don't expect you guys to remember all of this, but the main thing, if you remember, is that the PPP is divided into two phases. There's the non-oxidative phase, and then there's the oxidative phase. The non-ox is actually the more important part because we form the two most important parts. One of which is NADPH, remember, which provides the reducing power to the cell. The other one is the ribose 5-phosphate, which is important for nucleotide metabolism, like I mentioned many lectures ago. Now we'll see why this is the case. So your ribose 5-phosphate can actually be converted into phosphoribosal pyrophosphate, also known as 5-phosphoribosal 1-pyrophosphate, PRPP. And now PRPP is basically your sugar, your ribose, and it has these phosphate groups everywhere. So the only thing you need now is your base. And if you do the de novo pathway, you have to create the base on your own with amino acids. If you do the salvage pathway, the base will be there, so you just have to attach it on. And then this PRPP can be used for either purine or pyrimidine metabolism, and these both have separate pathways. Very important to note. Alright, so let's take a more in-depth look at that. So we see the PPP, pentose phosphate pathway, produces NADPH and ribose 5-phosphate. Remember, this is ribose. It has the OH, OH, and OH facing down, and it has the CH2OH up here. This ribose sugar will now undergo a reaction with the PRPP synthetase enzyme. Remember, synthetase has 3 syllables compared to synthase, which is 2. And we know that synthetase means that we use energy. So in this case, an ATP is used, and it gets converted to AMP, which means there are 2 phosphate residues coming off of it. So this will be added on to our ribose 5-phosphate. But wait, we already have a phosphate here. So instead, we'll be adding it on to carbon 1, the anomeric carbon. So we see here the pyrophosphate gets added on to the oxygen on the anomeric carbon. So we form 5 phospho, so this is the 5th carbon over here, so 5 phosphoribosal, this is the ribose sugar, and on this first carbon we have a pyrophosphate, so this is 1 pyrophosphate. So PRPP, 5 phosphoribosal 1 pyrophosphate. Hopefully that makes the name a bit easier to understand now. The other thing that you guys need to learn about is the nomenclature regarding this. So this is where it gets a bit tricky because honestly I didn't even realize this until learning it, because I always use these names like adenine and adenosine interchangeably, but they're not quite so. So remember, there are 3 main sort of layers. There's just the nitrogenous base, then there's the nucleoside, which is the nitrogenous base plus the sugar, and then there's the ribonucleotide monophosphate, which is the nucleoside plus a phosphate group. So for example, adenine is just the adenine base by itself, the 2 rings. Then if you say adenosine, that's adenine base plus the ribose sugar. And then adenylate, also known as AMP, is your adenosine nucleoside plus a phosphate group. And you can follow this through with the other 3 nucleotides. So just be very careful about the nomenclature. I'll try my best to get it proper, but I may mess up here or there. But yeah. So we'll be going over now Purine Biosynthesis and then Pyrimidine Biosynthesis. So I'll be covering the de novo pathway for purine as well as its salvage pathway. However, for pyrimidine, I'll only be covering its de novo pathway. So let's jump into purine synthesis, de novo. Remember, de novo means from scratch, so we don't have the base present. We'll be having to make it on our own. So remember, from the pentose phosphate pathway, we had our ribose 5-phosphate. Then it gets 2 phosphates added on to create 5-phosphoribosal 1 pyrophosphate, also known as PRPP. Now PRPP undergoes a 10-step reaction mechanism to form IMP. Thankfully, you guys don't have to know what goes on in these 10 steps. I do want to point out a few things though. We see glutamine, glycine, we see this, formiate, and this is just a misnomer for actually the THF form. So if you guys remember from those 3 biosynthetic fates of THF, one of them was purine metabolism. And this is the case here. So one of the formal THFs, well 2 of them actually, will come in and help convert PRPP into IMP, IMP. And then we also have aspartate and 4-ATP. So we see that IMP is actually called inosinate. So that's the nucleotide form. But IMP also has a base called hypoxanthine, and its nucleoside form, I believe, is called inosine. So now, once you form IMP, this is where the pathway will now bifurcate. So remember, this is purine metabolism. So the 2 purines are adenosine and guanosine. So at the bifurcation, we'll be forming adenosine on the left and guanosine on the right. Actually, sorry, adenylate and guanylate in this case because of the phosphate. So to form AMP, adenylate from IMP, which is inosinate. See, the nomenclature can be a bit tricky. You have to add a nitrogen on. So in this case, you're lacking a nitrogen on the hypoxanthine base found in IMP that adenine has. Remember, the adenine base, I'm not going to draw it properly, but it has this like flag nitrogen on the top. So you need to somehow add that on. So a common method of adding single nitrogens on in vivo is aspartate donation. So you add on an aspartate, and then in the second step, fumarate will come off. They don't show that, but fumarate comes off. Remember, which is a TCA cycle intermediate. And this net reaction of adding an aspartate and a fumarate leaving is 1 nitrogen being added to IMP. And this nitrogen converts IMP into AMP. And now your AMP, which is a ribonucleotide, remember it's not deoxy, it's ribo, so this is used in RNA. This can be phosphorylated from ADP or ATP. Now let's look at the other path in this bifurcation. So the IMP can also be converted to guanylate, which is GMP. The intermediate in that is xanthocene monophosphate. And so you can see that there's an intermediate in both pathways. And then this intermediate will be converted via ATP and glutamine into GMP. And once again, GMP can be phosphorylated into GDP and GTP. So let's take a quick look. So over here we saw our IMP. Remember, this is our hypoxanthine base over here. So in this hypoxanthine base, we actually don't have a nitrogen sticking up, we have an oxygen. Whereas if we look at our AMP, our adenine base, so remember, this is adenine, this is adenosine, and this is adenylate. So be careful of the nomenclature. But in this adenine base, we have a nitrogen sticking up. So somehow we need to convert this oxygen into a nitrogen. And the way we do that in vivo is through aspartate donation. So an aspartate will come in to form this adenylosuccinate intermediate, right? You don't have to really know the structure of that, but then just know a fumarate will pop off of it, and this will form AMP. And the net result of that is a nitrogen being added on. So that's how you form AMP. And remember, the IMP is called inosinate, its nucleoside is called inosine, and its base is called hypoxanthine. Whereas for AMP, the nucleotide is called adenylate, the nucleoside is called adenosine, and the base is called adenine. So it's quite a tongue twister. Alright, so now we just saw the de novo pathway, right? That was pretty easy. Now let's look at the salvage pathway. So remember, salvage is when the nucleotide bases are already present. So for example, one way this can be found is through our diet. So we ingest a lot of nucleic acids in our diet, and they'll be broken down from nucleic acids to nucleotides to nucleosides, which can then be taken up by our own cells. And these nucleosides can be turned into bases, and both these nucleosides and bases are sort of preformed. So we can immediately use them for the salvage pathway to form nucleotides, because that's a lot easier. However, if we don't use salvage, these guys will just be broken down into amino acid constituents and uric acid. Alright, so let's take a more in-depth look at what this salvage and degradation means. So remember, like I said before, we can uptake our nucleotides in our diet, and we'll be left with nucleosides and bases in our host cells, which can then be used to form nucleotides. So let's find this in this complicated diagram. So in this case, this adenosine, this inosine, and this guanosine are all nucleosides, which means that they're the sugar plus the base, and this is taken up by our diet. So in this case, yeah, these bases over here, they can be easily degraded into uric acid, which can then be peed out of our body. However, if we're really trying to make nucleotides, we can use these nucleosides and immediately form the nucleotide by adding on a phosphate. So in this case, for adenosine, we just need to add on a phosphate to form AMP. Same thing for inosine to IMP, and for guanosine into GMP. So that covers this part up here, the nucleoside. Remember, the other part of salvage is the base. So in this case, where are the bases in this diagram? So that would be adenine, hypoxanthine, and guanine. So to convert these guys to their nucleotide forms, we can also do that via this nucleoside kinase activity. I won't go too much in depth into it, but the main thing is your bases that are floating around can be converted into nucleotides. So that's salvage. However, there's also a degradation pathway, so these are very complementary. In this case, your degradation pathways will all end at uric acid. So let's start with AMP. Your AMP will cleave off a phosphate to form adenosine nucleoside. Your adenosine nucleoside will lose a nitrogen to form inosine. Your inosine, which is a nucleoside, will be converted into its hypoxanthine base by losing a sugar, and then this hypoxanthine will be converted into xanthine, and that'll be converted into uric acid. Similarly, your GMP will lose a phosphate to become guanosine, which is a nucleoside, so just the sugar plus the guanine base, and then this will lose the sugar to form the guanine base, and this will be converted to xanthine and eventually to uric acid. So we can see it's quite complicated. The one thing to note is this uric acid is actually what's responsible for the gout disease that people have. So normally it's excreted in urine, but high plasma levels of uric acid actually allow this uric acid to crystallize in our joints and the lining of it to form these gout crystals, which are very painful. Okay, so we covered a bit, so let's quickly summarize because that was pretty complicated. So normally from our pentose phosphate pathway, we form ribose 5-phosphate. and that gets converted into PRPP. This undergoes a 10-step reaction, part of which THF can come in and help out. Remember the formal THF. So now after these 10 steps, we form IMP, inosinate. So this is where the pathway bifurcates. So remember, we're working in de novo because we're doing this from scratch. So in this case, to form AMP, we need a nitrogen. So we do nitrogen donation. So we add on an aspartate and a fumarate comes off. This is how we form AMP. For GMP, we also have an intermediate, which is the XMP, your xanthocene. And from here, you'll form your GMP. So in this case, your de novo purine metabolism will convert our PRPP into AMP and GMP. And now to form the triphosphates, we'll just phosphorylate it twice into ADP and later ATP. And similarly, GDP and GTP. And now these nucleotides, GTP and ATP, can be used by your RNA polymerase to form RNA. So that's de novo. Now let's look at salvage. Remember, I told you two things can enter salvage. It can either be the base or it can be the nucleoside. So the nucleoside is just the base plus the sugar, so it needs a phosphate group added on. So in both of these cases, a phosphate is added. Whereas for adenine and guanine, you need the phosphate plus the sugar. So we can see here that this is the salvage pathway, when we already have the bases and the nucleosides pre-formed, whereas here we need 10 steps to actually make them. So it's a lot less expensive. And lastly, in green, I added degradation. So remember, there was that complicated degradation pathway in orange over here. But basically, we took AMP and GMP, and we converted it to uric acid, which, remember, is responsible for gout if it's present in too high of a concentration. Alright, so now let's look at de novo pyrimidine biosynthesis. So this one is a bit more interesting. So in this case, the first enzyme is called CAD. CAD is a trifunctional enzyme, which means it has 3 domains that catalyze the first 3 steps of pyrimidine biosynthesis. The C stands for carbamoyl phosphate synthetase 2. This should ring a bell since we covered CPS1 last time during the urea cycle. The A stands for aspartate transcarbamoylase. And the D stands for dihydroorotate. So these 3 components will be used to convert glutamine into dihydroorotate. And now this dihydroorotate can be oxidized to orotate, which then combines with PRPP, so that's this step over here. So this PRPP combines with orotate, and this forms OMP, which is then decarboxylated to form UMP. So you don't have to know these specific steps, just know that CAD catalyzes the first 3 reactions, and we see that amino acids are used in this. And then it undergoes a series of steps to form UMP, which is the uridine monophosphate. Now from here, similar to before where AMP and GMP can be phosphorylated twice, UMP can be phosphorylated twice to form UDP and UTP. And now you guys may be thinking, okay, this is one of the pyrimidines. Where's the other one? Where's the cytosine? Remember, because in purine, the pathway bifurcated to produce AMP and GMP. In this case, there's no bifurcation. So the UTP is actually a precursor to form cytosine. So you need to phosphorylate UMP twice to form UTP, which can then be converted to CTP via this glutamine reaction. And then this can be used for cytidine metabolism. So let's go back to that first enzyme in CAD, which was carbamoyl phosphate synthetase 2. I told you guys that this should ring some bells because we looked at CPS1 before. So if you remember, CPS1 was actually found in the mitochondria and was important for the urea cycle. So what it did was there was an enzyme that we used last time called glutamate dehydrogenase, which was a deaminating enzyme. So glutamate dehydrogenase would cleave off an amino group from our amino acid, and this would create free ammonia, which is not good for the cell. So something needs to pick that up. So CPS1, carbamoyl phosphate synthetase 1, will pick up free ammonia from the cell and use it to produce carbamoyl phosphate, which can then enter the urea cycle. However, in this case, we're looking at carbamoyl phosphate synthetase 2, and this is found in the cytosol And instead of using free ammonia produced by amino acid metabolism, we use glutamine as our nitrogen source, and this can be used to form carbamoyl phosphate synthetase. So that's the difference between these enzymes. So let's look at a comparison between the de novo pathways of purine and pyrimidine metabolism, because that was a bit confusing. So for purine, we start off with our PRPP, and it undergoes a 10-step reaction at least, sometimes 11, to form IMP, which is inosinate. And this is the building of the purine ring. So remember, in de novo, we actually have to build the ring ourselves, whereas in salvage, it's given to us. This IMP then bifurcates to produce AMP and GMP. Remember, there's an intermediate in between both steps. This AMP can then be phosphorylated twice to form ATP, and the GMP can be phosphorylated twice to form GTP. So these are your two purine ribonucleotides. So these can be used for RNA. They're not deoxy, they're just ribose. Now let's look at pyrimidine. So remember, for pyrimidine, we have CAD as our first three steps, and then we're going to create dihydroorotate, and then orotate, and then OMP. Then OMP combines with PRPP. Orotate combines with PRPP to form OMP, and this turns into UMP. Remember, you don't have to know all these steps, but just know that UMP is the first compound formed. And then UMP is phosphorylated twice into UTP. And this is important because whereas in purine synthesis, the pathway bifurcates, in pyrimidine it doesn't. So you need this UTP to form cytosine. So this last step is quite important to add in the extra nucleotide. So here's some questions. At what carbon does a deoxyribose sugar differ from a ribose sugar? Remember to pause the video because I'll be going through these quickly. So in this case, at carbon 2, a ribose sugar has an OH, which makes it labile. Remember, easily degradable. Whereas in deoxyribose, it lacks an OH, just as a hydrogen, and that makes it more stable, which is a good quality for DNA to have. How many high-energy phosphoanhydride bonds does it cost to make PRPP? So in this case, the answer is 2, because remember, we have ribose 5-phosphate from the pentose phosphate pathway, and the phosphate is on this 5th carbon. Remember, we need to add on a pyrophosphate to the first carbon, so we'll take our ATP, cleave off a pyrophosphate, and we're left with AMP. So we cleave off 2 phosphates. So that's how much this PRPP synthetase enzyme costs. Which pathway creates nucleotides from amino acids? A de novo pathway or a salvage pathway? That's right, a de novo pathway would, because this literally means from scratch. Where a salvage pathway, you're handed the base and you're handed the nucleoside, so it's much more easier to form the nucleotides. What is the name of the last intermediate before the de novo Purine Biosynthesis pathway bifurcates? Is it PRPP, IMP, AMP, or GMP? In this case, it is inosinate, IMP. Remember, we start off with PRPP, but it takes 10 steps to produce inosinate, which can then bifurcate to produce GMP and AMP. What is the name of the base in inosinate? Is it adenine, guanine, cytosine, thymine, xanthine, or hypoxanthine? Right, so this one is hypoxanthine. Remember, the corresponding nucleotides for these are, this is AMP, GMP, CMP, TMP, XMP, and this is IMP for this last one. It's just that the letter doesn't match up, right? You would expect this to be HMP. That's not it. This is IMP. So just keep that in mind. What is the end product of purine degradation? Not salvage, not de novo, but degradation. Is it inosine, xanthosine, hypoxanthine, xanthine, or uric acid? Remember, this one is uric acid because in this case, we have our AMP and our GMP, and they both follow this degradative pathway to form uric acid in orange. And uric acid is normally excreted in the urine, but if it's found in high plasma levels, it can crystallize, and this is the cause for gout. Okay, so let's summarize what we did so far. So in de novo purine synthesis, we created ATP and GTP. In de novo pyrimidine biosynthesis, we created UTP and CTP. What does this tell you, though? This is something interesting. One is we're missing one nucleotide. We haven't formed TTP. We haven't formed thymine yet. And the other is that these four are only ribonucleotides. They're not deoxy, which means as of right now, we can only use them to make RNA. We can't use them to make DNA. So what does this tell us? This is a very important facet in this metabolism, is that our deoxyribonucleotides are produced from our ribonucleotides. So the ribonucleotides come first. So this is sort of a diagram that explains how we create deoxyribonucleotides from the ribonucleotides that we just saw. So let's start off with de novo purine biosynthesis. So we know that in 10 steps we usually produce IMP, pathway bifurcates to produce AMP and GMP, and this can get phosphorylated to form ATP and GTP. However, this stops at one phosphorylation only. So you're only left with ADP and GDP. So these guys will then react with an enzyme called ribonucleotide reductase. And this enzyme will convert our diphosphates into deoxy ADP and deoxy GDP. So the important thing to note about this enzyme is it only works on diphosphate nucleotides. It doesn't work on triphosphates. So that's why we can't have ATP or GTP enter. It has to be ADP and GDP. So what this does is it'll cleave off that hydroxyl on the second carbon of the ribosugar, so you're left with the deoxy forms. Similarly, for the de novo pyrimidine, remember we undergo the CAD and all those other steps to form UMP. UMP usually gets phosphorylated twice to form UTP, which is important for forming CTP, right, cytosine, because this pathway doesn't bifurcate, so you need to form cytosine from your uridine. However, for cytosine, which is CTP, to turn into a deoxynucleotide, it needs to lose a phosphate, so it becomes CDP. So now we're left with CDP and UDP, so they'll both react with ribonucleotide reductase, similarly to these guys, and you'll be left with DCDP and DUDP. So these three up here, your adenine, your guanine, and your cytosine, will all react with a kinase to add on the remaining phosphate group to form DATP, DGTP, and DCTP. And this can be used by your DNA polymerases to extend your DNA chain. However, the problem with DUDP is that uracil is not used in DNA. So this is dangerous. If this is incorporated, then there's a problem. So this can never be phosphorylated like these guys are to form DUTP. That's not okay. So to prevent that from happening, we actually dephosphorylate it, or we use a phosphatase. So we form DUMP, dump. And dump needs to be converted to thymine, which is the nucleotide that's actually found in DNA. So there's a specific enzyme that converts our uracil to our thymine, and that's thymidylate synthase. So dump will be converted into DTMP, which can then be phosphorylated twice to form DTTP, which is then used by DNA polymerase to extend the DNA chain. Alright, so remember, the one thing to note is that uracil and thymine are both pyrimidines, and they both have this carbonyl sticking up and to the bottom left. The only difference is that thymine has this methyl group over here. The interesting thing, though, is in this last unit, though, we learned about one carbon carriers, so maybe that's the way we add on this methyl group. So let's see what happens. So remember, in de novo pyrimidine biosynthesis, we produce UMP, and UMP gets converted to UDP, and UDP can then be acted upon by ribonucleotide reductase. Remember, this enzyme only works on nucleotides with 2 phosphate groups. It can't work on ones with 3 or 1, only 2. So with this UDP, uridine diphosphate, our ribonucleotide reductase will cleave off the OH on the second carbon to produce deoxy uridine diphosphate, DUDP. But remember, this is bad, because now we have a deoxynucleotide, which is good for DNA, but this is a uridine, which is not found in DNA. So you cannot phosphorylate this, otherwise it can end up in DNA and mess everything up. So instead we actually have a phosphatase that'll convert it to dump, right? We take off a phosphate, and there's this enzyme called thymidylate synthase, which will actually add on this methyl group to create DTMP, which then gets phosphorylated twice and used by DNA polymerase to extend the chain. So the main thing we want to see now is how does this thymidylate synthase work? So let's start with our THF, our one carbon carrier, because remember, the difference between uracil and thymine is this one methyl over here, so we need something to donate that one carbon. So our THF will react with serine via serine hydroxymethyltransferase, that should be familiar now, and we produce the form N5N10 methylene THF. Now this form can donate a carbon to uracil, so it's going to donate this one over here, and that's going to form DTMP through the thymidylate synthase enzyme. Now in this process, this compound will lose a carbon, so it'll be converted to dihydrofolate, not to THF, but to dihydrofolate due to the oxidation state. So now it needs to somehow be reverted back to THF. So we need to reduce it one more time. So remember that enzyme, dihydrofolate reductase, which uses NADPH as a cofactor, will reduce dihydrofolate into THF. And remember, methotrexate is an inhibitor of this enzyme, which is why it's very useful for cancer therapies. Because if you inhibit this enzyme, now you can't produce thymine. This entire cycle stops. And if you don't have thymine, your cancer cells can't replicate their genome. So this is sort of the premise of why that occurs. Alright, so this is actually a pretty cool diagram I found on someone's Quizlet online, and it's pretty useful. It covers almost everything except for the salvage pathways. So let's quickly see what happened. So for Purine Biosynthesis, we start off with our ribose 5-phosphate from the Pentose phosphate pathway, converted to PRPP. PRPP undergoes a 10-step reaction, and remember we use amino acids, so glycine, glutamine, aspartate, but we also use our one-carbon carriers, N10-formyl and N10-formyl. So these guys are used to produce purines. So in these 10 steps, we produce inosinate, IMP. So this is where the pathway bifurcates. So to form AMP, we need to do nitrogen donation. So an aspartate will come in, and a fumarate will leave. And the net result of that is we produce adenylate, AMP, which is a nucleotide. Similarly, on the right side of this pathway, IMP will undergo two reactions to form GMP, which is guanylate. So now, these guys can be phosphorylated twice. So AMP can be turned into ADP, and then later ATP. Whereas GMP, similarly, can be converted into GDP and GTP. And both GTP and ATP that we just produced are ribonucleotides, so they can be used to produce RNA. However, what do we do if we need to produce DNA? Well, remember, our ribonucleotide reductase enzyme only works on diphosphates. So we take the ADP form and GDP form and convert it to deoxy-ADP and deoxy-GDP. And this is done by cleaving off the 2' hydroxyl group. And now we phosphorylate these guys one more time, and they can enter to form DNA. Now let's look at pyrimidine one last time, because I know this is beating it to death, but just to really sink in the concept. So remember, we start off with our CAD enzyme, which has 3 parts. It has the CPS2. Remember, that was the difference between that and the urea cycle. So it undergoes a bunch of steps. Eventually, it forms orotate, which combines with PRPP to form, I believe it was OMP, and then OMP gets decarboxylated to form UMP. So that's the main thing to know. UMP is produced. UMP gets phosphorylated twice into UTP, which can then be converted to CTP. Remember, UTP is required for cytosine formation. So the UTP can directly enter RNA metabolism, and so can the CTP. However, if we want to produce DNA, remember we need to work with diphosphates, because that's what our ribonucleotide reductase enzyme needs. So we'll work with UDP and convert it to DUDP, and for CDP, we'll convert it to deoxy-CDP. So this deoxy-CDP will get phosphorylated one last time to deoxy-CTP, which can then enter into DNA. But remember, your DUDP has a problem because it's not found in DNA. You need to form a thymine. So we can't phosphorylate it like we did to DCDP. We need to actually dephosphorylate it. So a phosphate group will come off and we form DUMP. And now dump needs a methyl group to be added on to form thymine. So we have this sort of thymidylate synthase enzyme, which takes N5N10 methylene, THF, it donates a carbon, and dihydrofolate will be produced. Dihydrofolate is then reduced to THF, and the whole cycle continues. In this process, DTMP is produced, and this gets phosphorylated twice to form DTTP, which can then be used to form DNA. Alright, so that was probably a lot, so let's just answer a few questions and we should be done. So what does ribonucleotide reductase act upon? Is it a nucleotide monophosphate, nucleotide diphosphate, nucleotide triphosphate, deoxynucleotide monophosphate, deoxynucleotide diphosphate, or deoxynucleotide triphosphate? The answer to this one is NDP. It usually acts on nucleotide diphosphates and converts it to a deoxynucleotide diphosphate. What form of THF is used by thymidylate synthase? In this case, it's N5N10 methylene THF. And remember, this is the form produced by the serine hydroxymethyltransferase enzyme. What is the substrate of thymidylate synthase? Is it UMP, UDP, UTP, DUMP, DUDP, or DUTP? This one is DUMP. Remember, the DUMP, your DUDP, which is produced, gets dephosphorylated first, and then this is converted to DTMP via the thymidylate synthase enzyme, and then this gets phosphorylated twice to form a deoxynucleotide triphosphate. Alright, and then lastly, I found this cool diagram online. I'm not going to go through it because I've pretty much beaten nucleotide metabolism to death. But if you want another angle at it, because it is a bit confusing, this is a cool diagram. I found it off of someone else's YouTube video, so it's not mine. But yeah, so this is all I have to cover today, so I hope you guys enjoyed the lecture. Thanks, everyone.